Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Kelly Flook from Thermo Fisher Scientific.
Good morning. Thank you. So welcome. Today we're going to be talking about a new resin from Thermo Fisher Scientific. This is the porous oligo DT25, designed specifically for scalable purification of in vitro transcribed mRNA. And as many of you may have noticed, there have been a lot of news around a global pandemic right now. And one of those vaccines that's a candidate to fight the COVID-19 is based on mRNA. So very exciting product, very exciting topic these days. And globally, as scientists, we're all banded together looking at diagnostics, treatments, and especially vaccines. And as you break down those vaccines, there's close to 200 molecules being assessed right now in this space. And what's really interesting, really exciting is when I first started looking at these numbers back in kind of March, April, the number of RNA candidates that were under consideration and moving into the clinic was less than 10. And to see now that number jump to 17, and I also feel like this slide is constantly out of date. So at the time I put this together, there were 17. It's really, really good to see the expansion of this technology as we look at trying to find a cure for this devastating virus. And across Thermo Fisher, we offer a broad range of products and services to fit your needs within the production space. And today, we're really just going to focus on purification and purification within our porous chromatography resin line. So as we look a little bit more about RNA, the nice thing about this molecule is that it's very versatile. It's essentially that computer code, right? So you're programming your body to develop and produce a protein for whatever you need it to do, which means it has utility in vaccines and therapies for oncology, protein replacement therapies, and as we're aware of right now, looking at ways to vaccinate against both common and rare infectious diseases. And then you couple together not just the challenges of manufacturing RNA. We've been making RNA for many, many years, primarily at a research scale. But now, as we try and figure out how we deliver millions of doses of vaccine to populations, how do we then manage that scale up? And this is a challenge that is really common across some of our customers. And as we develop this resin, we work closely with our customers, including Amptech, who are a leading RNA CMO in Europe. And they were tasked with developing a scalable and efficient solution to enable them to manufacture large amounts of messenger RNA, which would enable them to take on large-scale vaccine manufacture, including a vaccine for COVID-19. And as we looked through their challenges, really what we saw was they were primarily using reverse phase, which itself uses a lot of solvent. There's challenges with scale up when you're looking at having to put that into a facility. And again, not using chromatography per se, but more a smaller volume spin column format. So we really worked closely with them as they evaluated the porous oligo DT into their platform. So a little bit about the resin itself. So this is based on our porous resin technology. So this is a styrene divinyl benzene base bead coated with a hydrophilic coating to reduce nonspecific binding, and then the ligand that's attached to the surface is a 25-mer poly-T. And what this does is then selectively bind to that poly-A tail of the mRNA. And the nice thing about this is it makes the use of the resin really simple and really easy. You can add some salt if it's not already present to your IVT mixture. And then that salt neutralizes the backbone of the mRNA and the poly-T, which causes the hydrogen bonding to occur. At that point, you're flushing away all of your constituents, all of your IVT mixture components. Drop out the salt, the charge reappears, the hydrogen bonding breaks, and you release the purified poly-A containing mRNA. And from a binding capacity perspective, we've seen customers achieving up to 5 mg per mil for a 4,000 base pair mRNA, which is significantly higher to what you might see with some of the research products right now. And in many, many cases where we have excellent recoveries, greater than 90%, in most cases greater than 95, 96%. And again, this is based on a bead technology, which makes it scalable. So as you scale your process, you can scale your resin and your chromatography columns accordingly. So where does this fit in your platform process? So traditionally, you would use affinity purification at the start of your purification scheme. And in this case, as I mentioned, taking that IVT mixture, the only thing that will bind is something with a poly-A tail. So your DNA template, nucleotides, enzymes, buffer components, etc. Even a truncated sequence that's missing the tail won't bind to the resin. It will simply just flush through. In some cases, though, you may find that you have a product-related impurity, such as double-stranded RNA, or maybe an uncapped species that has the poly-A tail present, but it's undesirable. These may also be things like secondary structures. You may or may not want those present. In that case, we would recommend that you add a secondary polishing step with, say, ion exchange or HIC. One of the other unique ways that you can use the oligo-DT, and not saying that this is necessary, but it's a really good option should you choose it, is to use it downstream as either a final polishing step. So maybe you want to flip-flop say your HIC and your affinity, or in lieu of doing something like TFF, you can use it for buffer exchange. And like I said, you can elute in water, which means you can elute your molecule and then formulate directly from there. So from a binding capacity perspective, what you're looking at here on the left-hand side is a standardized experiment. So this is comparable conditions, not necessarily optimized, where we're looking at the binding capacity of 3 different sizes of RNA. And what you can see is that as the RNA size increases, you start to see a decrease in binding capacity. And this is what you would expect. A bigger molecule takes up more surface area, therefore you're likely to get less bound to the surface. Obviously, you can optimize your binding conditions in order to maximize that binding capacity. But in all cases on the right-hand side, you can see that independent of molecule size, independent of mRNA size, we don't see any impact in recovery. And you might be thinking, well, you do this with purified mRNA, that's all great. But if we do this straight from an IVT mixture, you can see in the graph on the left, we see no impact in recovery, whether you have those IVT components there or not. And this is really indicative to low nonspecific binding and the fact that the affinity approach means that just the polyadenylated species will bind. Then from a purity perspective, on the right-hand side, we're looking at the proteinaceous load. So this is primarily enzymes from your IVT mixture. And you can see in the flow-through portion labeled FT, that's where we see primarily the enzymes from our load solution. More importantly, you see nothing. There isn't anything detected within our elution peak. So as we take this into a purification scheme, you may be familiar with a standard purification chromatogram, where towards the beginning of the chromatogram during the load, we detect the nucleotides and the enzymes flowing through. And then after a washing step with slightly reduced salt, we then are eluting with water. We have a nice clean elution peak, followed by a base clean step, and then equilibration ready for the next run. So now if we look at the fractions from our elution peak, what you can see on the top panel is the reference unpurified IVT mixture. The peak at the beginning is your nucleotides, your proteins, your enzymes that don't bind to the reverse phase column. And then you can see those closely related impurities. So these are product-related impurities that are eluting very closely to that mRNA peak. And in the case of the IVT mixture, we're starting with close to 16% impurities as a factor of that total mRNA. Now in the middle panel, what you see is that with a reverse phase spin column purification method, we have a slight reduction in the product-related components. Now, all of our process-related components are basically pretty much not detected, but we do still have a significant amount of our product-related impurities remaining. Now, purifying with the oligo-DT, we start to see a significant reduction in those product-related impurities. And what this tells us is that we're really doing a much better job of removing those non-polyadenylated species from our mixture when we use an affinity resin compared to our the customer's reverse phase method. And right now we're working with our mass spectrometry team with some new software that they've developed to try and identify what those impurities are that are still remaining. And so with that, I hope that I've given you a good overview of the new porous oligo DT. And now I would like to thank you for your attention, and we can turn it over to questions. I'll do my best to answer them.
Okay, great. Thanks, Kelly. So do you observe a change in DBC for different length mRNA?
We do. As I mentioned earlier, the longer the mRNA, the bigger the molecular weight, we start to see a decrease in binding capacity. Now, we developed this resin to be designed primarily for up to 4,000 or 5,000 nucleotides. But as we start to see customers using more and more self-replicating mRNA that tend to be bigger on that kind of 8,000 to 11,000, we're still seeing binding capacities in the kind of 1 mg per mil range with some optimization of load conditions.
And what is the best way to prevent degradation of your mRNA as you purify it over porous oligo DT25 resin? And then the person notes that when they use larger mRNAs that they've generated using IVT, they always get consistent, very reproducible breakdown of RNA.
So if I think about some of the results that I've seen from our customers, I haven't seen any degradation of RNA happen during the purification step. In that case, customers are typically resolving it within that IVT process. If you are seeing it specifically during purification, then it may be the need to optimize some of your loading conditions and buffers to help stabilize the RNA. We can have one of our technical field specialists reach out to you and see if we can take a look at your issues.
And how do you determine titer in IVT mixture and then eluate to calculate the recovery?
So quantification in our RNA and oligonucleotides generally is challenging, right? we've seen customers have good success using a combination of different nucleotide detection kits. So, for example, looking at the quantity of RNA specifically, and then quantifying total nucleotides, total oligonucleotides versus double-stranded or single-stranded DNA, and then essentially having to back calculate. Other methods for quantitation kind of include using capillary electrophoresis, and then maybe HPLC as well. The DNA pack RP column that we use to do our purity analysis can also be used to do quantitation, because it will separate DNA from RNA.
And how do we clean the affinity resin, and do we see a decrease in DBC over reuse?
So we've tested cleaning cycles using a variety of different solvents and buffers. Typically, we're using anywhere between 0.2 and 0.5 molar sodium hydroxide as a kind of a cleaning strip step. If you want to or need to, it's also solvent stable as well. We haven't observed any decrease in ligand density as well as capacity, at least over kind of an initial kind of 10 to 20 cycles.
Okay, so if the elution was ssRNA and dsRNA, how can you separate the dsRNA from the ssRNA?
So yeah, in that case, as both of the RNAs have a poly-A tail, we recommend that you move on to a secondary polishing step, so something like HIC.
And what scale of mRNA can you purify with your largest VT25 column?
How big can you make a chromatography column? So it's scalable. So we have, within the porous line generally, we have customers doing, at least right now, RNA purification using, maybe a liter column to purify a couple of hundred micrograms of RNA. But you can pack this column into larger volumes. If you have enough RNA that you need to purify, you can certainly pack a liter column.
Okay, and which buffers do I need for the purification?
So we've seen really good success in just a simple 10 millimolar tris buffer with sodium chloride. You can use different additives. Customers are also using citrate. We find that works better than a TE buffer in some cases as well.
Okay, so we have time for just one more question. If we didn't get to your question, all of the questions, and there are a number of them, will be passed directly to Kelly, and she can follow up with you directly. So the last question is, do you heat the sample in sodium chloride before loading?
So you don't need to. I know a lot of research protocols or research purification protocols actually have baked into them a heating step to denature and then a heating step to elute. In this case, we've designed a resin to not need heat to aid elution. The resin is certainly stable to higher temperatures. So if you do need to heat because your construct is folding, or you need to break down higher order structures, you have the ability to do so, but it's not necessarily something that you have to do in order to use this column. Okay, So lots of excellent questions that are coming in. And yep, as mentioned, we'll get those questions over to me, and I will do my best to answer them and send our technical experts your way if I can't. And again, thank you for your attention. Excellent questions, and I look forward to getting in touch with some of you soon.
Thanks, Kelly. And as a reminder, the recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.